Hello, and a very warm welcome to everyone who has joined us today. As always, it's great to see so many people signing up for our webinars. And today we are presenting 3D ligand-based drug design. Understand binding conformations to identify and optimize novel active compounds. My name is John Norman. I'm Head of Marketing at Optibrium and moderator for today's webinar. And for today's webinar, I'm delighted to be joined by my colleagues, Peter Hunt, our Director of Computational Chemistry, and Matt Siegel, the CEO of Optibrium. And our special guest today is AJ Jain, founder and CEO of Biopharmix. So now it's time to hand over to our presenter, Peter Hunt, and host Matt Siegel from Optibrium, and to our guest, AJ Jain from Biopharmix. Matt, I will now hand things over to you.
Thank you very much for the introduction, John. It's great to see so many of you in the audience today. Thank you very much for joining us. My name's Matt Siegel. I'm the CEO here at Optibrium. And my main duty today is to introduce our two presenters and panelists. First of all, AJ Jain. He is founder and CEO of Biopharmix. AJ's worked in applied computer scientist in many domains. First of all, in the defense industry, continuing in speech and language processing, and ultimately leading to the bulk of his postgraduate career in computational chemistry and biology. Combining specialties in machine learning, drug discovery, and bioinformatics, AJ's current work is geared towards applications of computation to drug discovery, with an emphasis on making use of multiple methods and data types. He exploits synergies between protein structure data, chemical and target annotation information, and ligand binding affinity data. And we'll see the fruits of some of those labors in today's presentation. I'm also joined by Peter Hunt. So Peter is Director of Computational Chemistry at Optibrium. He joined us over 7 years ago from Novartis, where he led the UK CAD group, working in areas such as neuroscience, respiratory, and GI disease. Peter likes to describe himself as our in-house tame user of StarDrop, and as such was instrumental in guiding the development of the Surflex eSIM 3D module that you'll see today. Now, this webinar marks the launch of the new Surflex eSIM 3D module for StarDrop. It's an exciting culmination of over 2 years of collaboration with AJ and Anne. AJ, it's been an absolute pleasure to work with you and Anne. Just before we get started with the main presentation, maybe you could just share a little bit of background on how you came to develop the underlying methods and your Surflex platform.
Sure, yes, thank you, Matt. My background's a little bit unusual for somebody in Comp Chem, at least at the time that I got into the field. My undergraduate training was in biochemistry and computer science. I got my PhD in computer science at Carnegie Mellon University, and ended up in biotech primarily because the Cold War ended in the late 80s. And so after joining Eris Pharmaceutical in the very early 1990s, the idea was to try to bring ideas from computer science that were not well represented in Comp Chem into the field, beginning with the idea of looking at molecules from their surfaces as opposed to treating them as sort of atomic skeletons. So that nugget of an idea found its way into everything that we've done, whether it be docking or molecular similarity or predicting binding affinities. And over the last few years, it's become critical for calculational methods to become very fast, because the size of the screening libraries that people have are not huge. It used to be a few 100,000 compounds. Now it's several million, even for tractable available libraries, and up into the billions for larger ones. So we worked very hard to come up with the eSIM 3D method, both from the perspective of conformational search, but also especially for application to large databases where you can go through many molecules very quickly at speeds of hundreds of molecules a second on a single processing core. So a lot of hardcore computer science, a lot of knowledge of sort of protein and ligand interactions, all boiled down into a concentrated effort to make a real tool that people can use on a desktop or a laptop.
Thanks, AJ. I mean, the results have been absolutely amazing. I'd encourage our audience, if they haven't already, to go back and listen to the webinar that you gave on the underlying science when we first announced our collaboration back in the spring. So thank you very much. And without further ado, I think I'm going to hand over to Peter, and Peter's going to take us through the presentation and demo.
Welcome everybody to today's webinar. What I wanted to go through today was really an example of using eSIM 3D, the alignment generation, the creation of a hypothesis in terms of binding modes, and apply all of those to a virtual screening exercise. So that's the purpose of today, but hopefully you'll see from that all of the methodology that we have implemented within the new module. With respect to ligand-based alignments, there's been an awful lot of excitement recently with respect to the AlphaBold database, the creation of 3D structures purely from sequence, and the results of that have been quite remarkable. And the question would come, well, considering we can take a sequence and we can get a 3D structure, why don't we just do structure-based drug design? Why should we be doing ligand alignments at all? And I still think that ligand alignments have a very valuable place in drug discovery. With such generations of 3D structures, how does that structure compare to the protein that you have within your assay media? Is it in the right state? I mean, I know from personal experience that proteins move. Induced fit is a real and most definite phenomenon. I see this from my experience with HIV protease, which is why I illustrate that on the right-hand side. The first crystal structure that came out was the APO form, and in red, as shown here, you can see the backbone trace, a wonderfully large active site, and we started designing compounds to that active site, and they didn't work. And it was only when the first inhibitors were co-crystallized that we could see just how dramatic the induced fit with that flat movement was in the purple structure here. And then that's when our design efforts started working. So if you happen to have an APO structure and it's a nice novel protein, A, do you know it's in the right state? B, where is the pocket? You have to look at pocket finding algorithms, guesswork to see where such things may bind. And the final point really is a practical one. We're going a lot more into phenotypic screening these days. And in that particular case, how do you know what your target is? Therefore, how do you know what you can do a structure-based design method on? However, what you do know is that you have small molecules that work. And what you hope is that they work by the same mechanism, and therefore the combination of those active ligands will help you infer the nature of the target binding site that you're dealing with. So you can still progress because you have ligands that work. And indeed, AJ and Anne have published quite recently their work on the combination of ligand and structure-based design, and this actually produces even better results in terms of the ability and the understanding for drug discovery. Hypothesis generation. If you happen to have those ligands that work, and you can combine those and bring them in to create a consistent set of potential binding modes. I like to think of it in terms of a Picasso painting. He strove to give the viewer various aspects of their subject to create something more than just a simple painting. And I like to think with respect to the combination of ligands, you are bringing in different facets of the binding site, the target binding site, and it is that combination of those facets which gives you the most information. And so what we can do within the eSIM module is to take a set of 2D ligands, generate the range of possible conformations for those, compare those combinations and combine them such that we can create a consistent view or potential view of the binding conformations. And we're able to generate surfaces from there, looking at the structural properties of those molecules to be able to say, well, these are the areas of consistency. These are the areas where really you probably shouldn't be changing, but what are the features that are really only common to a few of these ligands? Where can you transfer features and SAR from one series to another? And you can get that with a concerned overlay of those new ligands. To get that all to work, we need to have good conformational sampling. And with this, we use the Biopharmics ForceGen routine. It is published, and there are references you see at the bottom of the slides here. ForceGen is faster and more accurate in the benchmark tests than any other system. Underlying it, we have the Merck molecular force field, or at least a variation of MMF94, and we provide different levels of sampling to suit the need that you have. If you're looking at virtual screening, then you're probably going to have lots of molecules, and really only you want to do is to prioritize those, bring them to the top, which is the task we're going to look at today. And then in that particular case, you probably don't want to have a huge amount of conformational sampling because that fine detail is not necessary. However, if you happen to have molecules that are highly flexible, contain lots of rings, or you want to have more accurate alignments and posings within proteins, then again, you have that capability of having a more detailed conformational sampling. And also, just as a nice point, if you happen to have epimers, diastereomers within your set, then we can enumerate all the possibilities for you, for your requirements, and be able to investigate whether you've got the better epimer or the best diastereomer in your representation. So you can come in with racemates, we can explore those different possibilities for you. Also within the sampling, we do have the ability to put positional and torsional constraints. If there are areas, perhaps a side chain that you know is going to go into solvent, it doesn't really make much sense to explore all of the conformational freedom of that side chain. Therefore, you can constrain it and limit the conformational exploration to the areas that you consider to be important. And again, as I deal with some of the university courses that I give, actually having a consistent charge state for your compounds, if we're dealing with ligand-based alignment, is key. And so we have a heuristic here for protonation, deprotonation of groups, such that you can treat these groups consistently throughout the whole process. There's no good trying to take a protonated amine and then try and find its equivalent in a neutral amine, because the charge states are just going to be too different. So how does this all work? We look at a particular reference ligand and we surround it with observer points. And these observer points are brought in close to the ligand, and then the density of those points is lessened to a particular value that we provide. And at each of those observer points, we look at 6 properties: steric and coulombic interactions, as well as the distance to hydrogen bond donors and acceptors, and the angle that obtains from the observer point through that hydrogen bond. And with these properties, if we move from this 2D representation, just to show you what happens, and move it to a more realistic 3D representation here of our reference ligands to pyrrolidone, you can see that we can actually compare the shape similarities from these alignments for various molecules, and then come up with an alignment score between 0 and 1, very much the similarity values here. And we can see that the cyclopentanone has, again, very good shape correspondence. It has the hydrogen bonding accepting correspondence to pyrrolidone. It will just lack the area and the observer points for this hydrogen bonding. And therefore, it's not a perfect match, but it is better than the other examples that we have here. And we can quantify that with the similarities. So the demonstration today is going to be a virtual screening exercise. And so what I hope to do within this is to work through those steps of aligning of compounds to a single ligand. This could just be a simple case that you want to align two molecules. You've got a bioactive conformation that you know about. How does another molecule work? So we can go through the alignments. You can assess those individual alignments to see where things can be transferred between one series and another and the consistencies between series. And then we'll create a multi-ligand alignment. Again, we can see the effect of aligning the large set of compounds to that multi-ligand alignment and to compare it to the single ligand alignment and show you how you can effectively differentiate actives from decoys. Now, the way that we're going to be doing this is by using the statistics of area under curve from a rock plot here. This is a plot of true positive rate versus false positive rate. And essentially, if we get an ordering of all the positives before all the negatives, actives before the decoys, we would have a curve that would go up here and across, and the area under that curve would be 1. That's perfect differentiation. If we were random, then we would find actives at the same rate as decoys, and therefore we would follow this path. And of course, the area under that curve is a half. So what we're looking for are area or values close to 1 and certainly not at 0.5. So this is the area that we will be describing for you as our means of success in a virtual screen. The dataset is from the DUD-E dataset. These are decoys and actives that have been extracted from Kemble for the poly-ADP ribose polymerase, part 1. And really, this is just to say that we got 3,000 decoys, 500 actives, and as was the design, this is just to show you that simple phys-chem properties, there really is no differentiation between these two sets. And so with that, I will come out of the presentation and I will go into StarDrop. And so I've just opened this particular data set, and here you can see that I have a dashboard, and these are basically the plots that I've just shown you before with the distributions of this virtual screening set compared to actives and decoys. The data set itself has 3 tabs in it. The first is just a small sample. This is in fact a set of actives and decoys, which are only 150 of them. And this is really just for time purposes because we are dealing with a webinar. We do have the full virtual screen information here, and we have a library of 5 compounds that are all actives. We have one of those compounds that actually has a conformation, a bioactive conformation for it, and we will use that as our first alignment. With this small set of actives and decoys, I am going to be comparing and aligning this to 2 different reference sets. So a single reference ligand and a multiple alignment. And then in that case, a lot of the time it takes to do these type of analyses is taken up in the conformer generation itself. And so what I've done here is that I've actually generated the conformations of molecules beforehand. And if I show you with this particular row, if I highlight it, it pops it into our 3D window here, and I can look at the 34 conformations that have been generated for this particular molecule simply by clicking and going down and surveying. I can actually, in fact, compare conformations. This is not particularly flexible molecule. There are only a few rotatable bonds in this particular case, but you can select rows and be able to see the conformational freedom for any particular molecule. So what I'm going to do is to highlight all of the compounds in this data set, and I shall start the process of alignment using the blue button down the bottom. And here I can calculate alignment similarity. Just to highlight the fact, this is where I would also, if I were starting from scratch, I would highlight the rows and generate the conformations. And in this particular process, I can decide exactly what are the sorts of conformational freedoms and specificities and ring explorations, etc., that I want. So I can put in whatever I want in terms of that definition for our conformational sampling. But in this case, I'm just going to calculate the alignment similarity, and it asked me for a reference. Now, I can open up a simple file on this, or I can download a particular bioactive conformation from a PDB. But in this particular case, I happen to have that conformation already there in my actives set. And so I'm going to select that row, and you can see that now gets added to the 3D tab. I can select the molecule, and once it's selected in the 3D tab, I now can add it using this button as the reference. I'm going back to my set. I will give this a name. So align OK, and I'll hit next. And at this particular case, again, for some sort of speed, I'm going to have the fastest alignment. But again, I've got multiple options here available to me. And I'm going to hit finish. And you can see that very, very quickly, I have got alignment similarities for all of this data set. So 150 molecules complete within, what, second and a half, 2 seconds? Now, before I analyze and have a look at these alignments, what I'm going to do is I'm going to kick off the hypothesis generation. So I've gone back to that set of actives, and I will select all of those actives, go to the blue button and say generate binding hypotheses. Again, in this case, just for speed, I'm going to go for the fastest. I'm going to hit finish. When we do this, we can see that we start the process running, and with this we need to generate all of the conformations. We need to then combine those conformations pairwise and add in all of the particular ligands and find 10 hypotheses there you get the best overlaps between those 5 compounds. When that finishes, it should pop up. But for now, what I'm going to do is just have a look at the alignment for, say, this particular molecule with one WOK. So in this case, I can go back to my conformer list, and I can see that I've got eSIM alignments here. I can just select that, and I can have a look at a variety of the alignments. The reference is in green. This is 1WOK, it is in green. And my row, a particular alignment, I can just select and see how it compares. And you can see the alignment similarity scores here on the right-hand side. As I showed before, we have the ability to look at the properties of these and compare them. And so if I click on this gear button here, I can show the surface, and one can see the similarity in various properties, either the sterics, and the hypothesis has finished. So in this case, I'm going to take off the sterics, and go back to hydrogen bond donors and acceptors, and you can see in this particular case, here is the similarity surfaces. This is where there's consistent binding, and you can see just how much we've got this hydrogen bond donor and acceptor patches here. There's quite a patch for hydrogen bond acceptors, but it's not universal. There's a little part for a hydrogen bond donor here we're missing, but there are a couple of hydrogen bond donor points up here, but you can just see the consistency in this warhead area as opposed to the tail region. And this is the way you can look at the different hypotheses and just see how those different alignments, we have a probability that's associated with each of these hypotheses. That probability is affected by ligand strain. So you can see that the components here have different amounts of strain to get into this particular alignment. That plays a part in the probabilities. And again, you can just flick through these and find which ones that meets your particular view of how you think these compounds are binding. So you can bring your own knowledge into this as well. But in this case, we're just going to deal with hypothesis number 1. That's the most probable, and we use that for an alignment. Just going back to this particular set of actives and decoys, and just a nice little feature with respect to the display. If I go back to the alignment of this molecule with the reference, there we go. When we look at the surface, there is the possibility of viewing side by side. And so again, one can see the reference and your aligned ligand, be able to see those similarities, but you can also see the differences. And so the alignment is not perfect around that amide, and so this is why you see differences in this region, because it's not quite aligned. You have a most definite difference here in the sense of my reference has a hydrogen bond acceptor and it's not present in my new molecule. And just my new molecule has the hydrogen bond donor here, the charged basic amine, and that's not present in my reference. So nice to be able to play with these visualizations to be able to provide you with the information of what is consistent and what is not between various chemotypes. So I'm just going to take those surfaces off, and now we're going to do the alignment to the multiple ligand, the hypothesis. So again, I just select the rows, go to the blue button, calculate alignment similarity. I already have one alignment here, and if I have aligned a few molecules to it, say, but then I want to align some more, it's very easy to do that same alignment because I could just choose that column and all of the parameters that I have to create an alignment will carry through. But in this case, I want to define a new alignment. I'm going to use a new set of reference ligands. And so I tell it I want to define a new reference, and I get to the selection. Now you can see that I have a hypothesis that's available to me, and so this button is now active, and so I can select it, look at the various hypotheses. As I say, you can remind yourself what the hypotheses are, but I'm just going to choose the top one and hit OK, and that gets entered as a new reference. I'm going to give this another name, align multi, next, and again I'll choose the fastest and hit finish. Now I'm comparing all the conformations of my molecules to that combination of 5 ligands in a hypothesis. So it's probably taken, what, another second longer than the previous one. And now we have generated 2 sets of alignment similarities to a single reference and to a multi-reference. And so I now need to go to the visualization tab here to be able to see, well, how effective have I been in this virtual screening exercise? I'm just going to go to the ROC curve and sample set. So that's the column that contains the actives and decoy definitions. I'm going to be interested in actives, and just because it's looking at the first column, happens to be ForceGen conformations, that's not particularly good, but then you wouldn't expect it to be. So we're going to select the single reference here, and you can see we've got an AUC of 0.78. That's pretty good, and you can see just how well we've started off. Perfect discrimination at the beginning, and then we tail up. So an excellent discrimination even with a single ligand. However, having multiple ligands, we now have an AUC of 0.92. Again, wonderful start, perfect discrimination here, and again, very good discrimination of actives versus decoys. But I can hear you screaming at your laptop saying, well, this is just a subset. This isn't, you could be biased. Well, this is why I have the output from the full virtual screen. And if we look again at the ROC plots for the sample set, again, we're looking at actives. In this case, we're looking at 3,000 decoys, 500 actives. We're doing pretty well with a single ligand. Again, very, very similar, 0.72 on the AUC. But if we select the multi-alignment, we do almost as well as we have seen in that small subset, an AUC of 0.9, an excellent discrimination at the beginning of this, and then curving off. And so the alignment of these molecules to that multiple ligand means that we have a much better assessment of those actives and decoys, how similar they are to known actives, and be able to properly assess those compounds as whether they're likely to be actives or not. And you can see just how well we do by using that multiple ligand alignment. And just because I showed you that you have those property values calculated, if I look at some of those properties and compare them, you can see we're getting no discrimination at all from here. I mean, this is exactly how this study set was designed. And so we can see it that we are really getting good results only when we're looking at that ligand alignment in this case. And so that is the demonstration. If I just go back to the slides, what I hope that you've been able to see today is that we can take any number of molecules really and align them to not only a single reference conformation and be able to examine the similarities and differences between the reference ligand and the aligned ligand. We can create a binding hypothesis, in fact, a number of hypotheses from a set of multiple ligands, and again, be able to assess the similarities, the complementarities, and the differences between each of those chemotypes, such that you can look at the features of one and be able to transfer it to another. And then using that multiple alignment, we get excellent discrimination in this particular example virtual screen to differentiate actives from property matched decoys. And with that, I thank you very much for your attention, and I will hand back to Matt.
Thank you very much, Peter, for a great presentation and demo. I think one of the things I would emphasize as well is that not only are we getting very good results from the use of eSIM3D, but we're getting those results for the right reasons. And this is something that our colleagues at Biopharmix have spent a long time looking at. And these visualizations that Peter described really help us to understand the reasons for those alignments and those similarities, and also help to guide further design, new opportunities to modify compounds and further improve their activities. And during the presentation, you'll have noticed several references go past that really demonstrate the very rigorous way that these methods have been validated, and would encourage you to look up those references to learn more. Or indeed, AJ Jain, who's the CEO and founder of Biopharmix, gave a great webinar earlier this spring when we first announced our collaboration, talking about all of the sort of underlying science and methodology behind it. And that webinar is available as a recording on our online community website.
And as an addition to that, Matt, the PARP1 example that I used to generate the binding hypotheses, those ligands that I used were actives because we knew that they already bound. They have crystal structures, and if one looks at the hypotheses that were generated versus the actual alignment from the crystal structures, the hypotheses generated are very, very good and close to the actual values that we see from the crystal structures. I could have used the crystal structure alignment today, but I wanted to illustrate how one could take a set of ligands and generate hypotheses within the tool.
That's a really good point, Peter. Thank you. So without further ado, we should move on to the Q&A session. So some great questions here. We'll start at the top. There was a really interesting question about the mutual alignments. The example certainly that you showed in the slides, Peter, mostly we're looking at sort of planar molecules, which I guess conceptually might be easier to align than sort of more aliphatic, higher sp3 molecules. I don't know whether Peter and perhaps AJ, you want to comment on the performance of eSIM with more flexible molecules.
Yeah, sure. It's a great question, and yeah, you see with the PARP ligands, very sort of flat, sort of aromatic molecules, particularly in the sort of warhead that binds deep in the active site. We've done really huge calculations on predicting the relative pose of molecules where it's known for crystallography. Almost 500,000 such comparisons, and the details are in the paper. Many of those involve molecule comparisons where you're looking at things that are more like natural products, so very 3D. So even in cases, for example, where one molecule is say a macrocycle and another one is a non-macrocycle, we get accurate predictions of pose, and this has been quantitatively measured and compared to other methods. So the underlying pose predictions are very accurate. We're in the process of writing up a paper on the effectiveness of multiple ligand alignment as opposed to the single pairwise pose prediction. I can't really share any quantitative results, but we have gone through and looked at many cases where what we would consider the molecules more interesting than what you see with PARP. We don't see any degradation of performance on things that are sort of more 3D than the molecules that we saw in the demo.
I think if I can add to that, it's the ability to be able to change those parameters for conformational sampling. That is something where the user is not restrained by having a particular set. And so if you do find yourself in a particular scenario where your compounds are highly flexible, you have the ability to be able to really fully explore that conformational freedom and be assured that you will be able to make good comparisons. Yes, in this particular set, we have limited rotational freedoms. They're not all aromatic, coplanar, because there's some there are some rotatable bonds in there as well. But this is a good example for showing just the speed and the ability to create those good alignments. But you have the flexibility, you have the control to be able to do the right thing with the molecules that you have.
Fantastic. While we're on the topic of mutual alignments, there's a great question. We showed an example of a multiple ligand alignment on 5 actives. What if I have 500? Can you do the same on 500, or is there a limit? Well, I'll just start by answering the first part of that myself, which is within StarDrop, to make the computational cost reasonable to run on the desktop, we do have a limit of a mutual alignment of 5 actives at any one time. But I know that in the full Surflex platform, AJ, you can align more molecules simultaneously. How does that cost scale in practice?
In practice, what we do in the fully automated method for doing a very large multiple ligand alignment is build up a smaller core hypothesis and then expand that. A user of StarDrop can approximate that same behavior by selecting, for example, 5 actives, ideally which have slightly different underlying scaffolding, because you get better absolute configurational predictions if you have somewhat different scaffolds. Generate a binding site hypothesis, as Peter showed how to do. Look through those. Now, the multiple ligand alignment takes into account conformational strain as well as the similarity of the ligands, and so as well as compactness overall volumetrically. There are several features that go into that. Once you find a multiple ligand alignment that you think comports with the SAR of a system, and usually that'll be within the top 2 or 3 of the alignments in the quality experiments that we've done to assess, then you can do an alignment of your remaining, say, 500 or 5,000 ligands to that using a deep conformer search and the highest quality eSIM alignment methods. You can build a multiple ligand alignment now of many hundreds of molecules very quickly, and also incorporate some of your own intuition about which alignments make the most sense given what else you know about a system.
That's great, thanks a lot. So we have a few sort of practical questions here. So first of all, maybe for you, Peter, can the ForceGen component be used independently?
Yes, I didn't actually demonstrate it, but I did mention at the beginning in terms of the ability to generate the conformations for any number of molecules that you have within your StarDrop session. And so again, you have that flexibility to allow complete sampling, or just to restrict some of the sampling with torsional constraints. So that's the ForceGen component. In the alignments and the hypothesis generation, obviously that is inbuilt in the whole process, and that executes under the hood before the alignment happens. But you can generate conformations with the ForceGen separately.
Excellent. And actually, a related question, does the alignment require X-ray coordinates, I assume for the reference, or can the actives be defined from ForceGen conformers?
Oh, certainly the actives can be defined by ForceGen conformers. In the example that I had of the actives, I had one compound which had a conformation, which was a conformation from a crystal structure, but it certainly does not have to be. And in that example, we actually took all of the molecules and generated a full conformational sampling with those, and it wasn't just a case of aligning the other 4 to the single X-ray conformation. So
yeah, the multiple ligand alignment that Peter showed came from de novo ForceGen conformations. It turns out if you were to actually look then at the comparison between that multiple ligand alignment that was purely ligand-based with no information from the crystal structures, if you compare that to the actual crystal structures aligned by the protein active sites, it turns out it's actually very, very close to correct. One odd thing is that there's 2 sort of distal amines on those ligands. It turns out in PARP1, they actually bind in 2 slightly different locations, both where there's a carboxylate available in the protein. So the alignment that's generated is very close to correct. In fact, almost absolutely correct in the heterocyclic portion of the molecules. But for one of the 5 ligands, it actually places the tail where I think a person would guess it should go, but it's actually wrong. That doesn't end up actually hurting the ability of eSIM3D to do extremely well in the virtual screen, whether you use the nominally correct crystallographic alignment or the multiple ligand alignment that Peter showed, you do extremely well. So multiple alignments don't have to be exactly correct in terms of the experimental data, and that's why the purely ligand-based approaches work very well.
Fantastic. So another practical question here. Can you display the protein too? One of our eagle-eyed audience spotted the protein drop-down in the interface.
Indeed, one can. And whilst I was displaying a single conformation for an active molecule, as I said, this was extracted from a protein prior to the demonstration, but you can do that extraction directly within StarDrop. The choosing of a reference, that can come straight from a PDB code and be able to extract the ligand from there, or you can download to the 3D viewer that we have and be able to select the particular crystallized ligand from that situation. So there are a number of options that one can use to choose the appropriate reference for any alignment.
Great. So one of the things that we didn't talk about was benchmarking. So maybe AJ, you could answer this one. How fast is the virtual screen? Can you share any statistics you have on this matter?
Yeah, so the paper that you'll all get a link to, it's open access, that we published in 2019, has extensive benchmarking data, a single core data, comparative performance against, I think, 7 other virtual screening methods, including probably the most widely used method, the ROX approach. What we show is that in the fastest mode of running eSIM3D, it's approximately 5 times faster than ROX, which proceeds at about 50 molecules per second. eSIM3D is about 274 molecules per second on a single core. Multiple core operation obviously goes faster, but it's definitely the fastest method out there that's not GPU based. But the more important aspect, I think, even apart from raw speed, is that at those speeds, it's performing quantitatively better than every other publicly benchmarked method for calculating 3D similarity for virtual screening. So you get both speed and accuracy. So that doesn't happen very often, and it hasn't happened very often in my lifetime. So we're very happy that this is a pretty sort of interactive speed type of algorithm.
That's great, AJ. So we've got a couple of really good scientific questions here as well. So AJ, can you comment on the role of energetics and Boltzmann population weighting?
Oh boy. Okay. So when ForceGen is doing conformational sampling, it's doing it in a non-stochastic way, always trying to move through the lowest energy sample of a conformer pool as a search is progressing. So ultimately, you might start out with a not very good initial 3D guess. It's not going to have terribly high strain, but it's not going to be perfect. As the ForceGen process proceeds, you end up getting a lower and lower energy window. Now, we don't try to do a Boltzmann sampling of the conformational space of a molecule. What we try to do is get a diverse structural sampling. So instead of, for example, trying to produce 250 non-redundant conformers, for example, some methods do that, we try to produce at the PGOM level 250 maximally different sort of well-spaced conformations within an energy window, which is typically set at 10 kcals per mole. By doing that, by explicitly seeking a diverse set of conformations, we're able to cover with higher probability the bioactive bound pose of molecules. This has been shown on Johannes Kirchmeyer's platinum diverse set, nearly 3,000 PDB ligands bound to proteins. So the strategic question in terms of conformer search that we wanted to address was reliably being able to find something close to the global minimum and then sampling the window close to that such that it was very likely that one could uncover the bioactive pose very close to how something would bind. So we return conformational minima that are well spaced that sample that energy window. If somebody wants to then take that pool and generate a Boltzmann weighted distribution, they can do that. But that isn't the goal that we had in mind. It's very much driven towards, can we generate high quality virtual screening conform databases, and can we accurately predict poses? Can we produce good multiple ligand alignments? Those questions aren't well served by a Boltzmann distribution, which would oversample the lowest energy conformers, which aren't exactly the ones that bind.
That makes sense. Thanks, AJ. So here's a question that perhaps gives us a bit of a glimpse of the future, and I think goes beyond eSIM 3D itself. But I know it's an area you've been working on very hard, AJ. So if you had activity data for a series of compounds, can that information be included along with structural alignments to improve the AUC?
So I guess the bigger answer is yes, we have technology to specifically model quantitative binding affinity data using a methodology that extends the eSIM approach to build weighted binding site models. And yes, you can absolutely take advantage of binding affinity data, build quantitative models, and those tend to be much more specific in terms of being able to pick out highly active molecules from less active ones, but also obviously decoys. That is not something as yet that we've integrated within StarDrop. It would seem like an obvious way to move in the future. We're actually about to submit a paper on the entire technology applied to a very large set of benchmarks where FEP Plus has been applied, and the results are really quite impressive. But yeah, binding affinity data can be used to build weighted models. It's not something that's in StarDrop right now. I hope that we can offer that in the future.
Yeah, absolutely. And if you want to know more about that, then AJ and Anne at Biopharmix will be very, very happy, I'm sure, to tell you more, and we'll be happy to put you in touch. And I guess the last really question here relates to sort of size of screening collection and how many molecules you can screen. There's a few of them around that. And I think here it's also worth drawing a little bit of a distinction between the eSIM 3D and module in StarDrop and the full Surflex platform that our colleagues at Biopharmix provide. StarDrop, the eSIM 3D module in StarDrop is designed as a desktop tool and running on the desktop just to keep sort of sane limits. We use a maximum of 8 cores simultaneously. And I guess the ultimate limit really is that the amount of memory you have for storing and processing sort of the data sets with potentially large numbers of 3D conformations. So you can certainly get well into the thousands, probably tens of thousands of compounds on a good laptop, or maybe a bit higher on a high power workstation. And that's really sort of the limitation is the memory really from StarDrop's perspective. But with the full Surflex platform that Biopharmix produce, then I think the sky's the limit. Is that fair to say, AJ?
Yeah, it is. Thank you, Matt. We have people routinely running 2 and 3 million compound screens with the Surflex platform. And one thing we could do is pre-calculate the databases for, say, enamine real or the zinc lead-like, and it would not be hard to extend the StarDrop interface to provide users with a pull down to say, hey, can I screen Enneman reel against these 2.1 against this particular binding site hypothesis or crystal structure. It would require some engineering for StarDrop to go in and pick out the top scoring things so that the interface doesn't get overwhelmed by having to try to visualize a 2.1 million row table. But that's the kind of thing that if people are interested, it's certainly doable. I'm putting a little bit of a few words, I guess, into Matt's mouth, but it's something that you know, from a technical perspective, is certainly doable, and it'd be very easy for us to share pre-calculated libraries with people as well.
Yeah, that would be a really great combination. You're absolutely right, AJ. Well, with impeccable timing, we have reached the end of our hour. So that really remains for me to say a big thank you to everyone for joining us today. And last of all, a big thank you for joining us for today's webinar. I hope you found it useful and interesting. If you do want any information, would like to arrange a trial of eSIM 3D within StarDrop, and have any other questions, please do contact us at info@optibrium.com. And as John said right at the beginning, this webinar has been recorded and it will be available on our online community at optibrium.com/community as soon as possible. We'll send you a link, and you're very welcome to come back and refresh your memory, or indeed share the link with your colleagues. So thank you again for joining us.